Three well-known examples of multicellularity includes animals, fungi, and land plants. The multicellularity is characterized with a complex development, cell differentiation, and tissue formation. Interestingly, in fungi, there are multiple secondary reversion to the unicellular state. For example, yeasts of the model genus Saccharomyces, the well-known organism. The case of such reversals in animals are, for example, Myxosporidia, the parasitic cnidaria almost reversed to unicellular conditions, and canine transmissible tumor, which is caused by dog-owned cells reversed to unicellular lifestyle, and parasitic condition. The complex multicellularity, which includes macroscopic size of the organism, high level of integrity, cell and tissue differentiation, was also independently acquired within brown and red seaweeds, Heteroconta. But there are much more cases of different independent variants of multicellularity in protists in all major clades, which express different levels of complexity, integrity, differentiation of the cells. And here we will review some selected examples of this diversity. Not to be confused, we need to distinguish between clonal multicellularity, when the multicellular organism consists of cells originated throughout divisions of the single initial cell, and aggregative multicellularity, when the multicellular organism is the product of aggregation of cells, which used to be independent single-celled organisms. The simplest type of multicellular condition in protists is colony. Colonies can be small and include only several cells, or huge with thousands of cells and macroscopic size, like in green algae from the genus Hydrodiction. The cells in the colony can be connected by skeletal parts, like in golden algal genus Dinobrion, or the cells can be embedded into a common mucilage matrix, like, for example, in the haptophyte Phaeocystis. The most complex colonies are known in Pyrithric ciliates Zootamnium and green algae Volvox. Zootamnium forms a giant colonies up to 15 millimeters, attached to the substratum via branching stalk, with smaller cells on the branch tips and larger cells at the bases of each branch. Larger cells can form swarmers with the cells detaching from the colony and settle at some place to form a new colony. The smaller cells are specialized in feeding and never give rise to the new colonies. The situation is reminiscent of separation to soma and germline in complex multicellular organisms, where only a particular type of cells give rise to the next generation. The same is true for colonies of Volvox, where the germline is separated from somatic cells, propelling the colony through the water column via flagella. The colonies of Volvox are similar to complex multicellular organisms in many ways. The colony is polarized and swims towards the light. The cells in the wall of the spherical colony are different in size, gradually getting smaller towards the posterior end, and also having similar size gradient of the eye spots. Large reproductive cells are located inside the sphere and do not take part in the locomotion. At some point, they start to differentiate into new colonies undergoing a series of mitotic divisions without growth, which is very similar to the cleavage in multicellular organisms. This similarity is enforced by the remarkable process of inversion, when the cell layer, which initially is facing the flagella side, get inverted inside out, with future generative cells taking its position inside and the flagella outside. This process is similar to gastrulation in metazoan embryos. Aggregative multicellularity is known to emerge independently in all eukaryotic lineages except for Archaeplastida, and also in eubacteria, in the example of myxobacteria. Here we will review the best studied example, the amoebozoan genus Dictyostelium. The microscopic amoeba live in the soil as independent unicellular organisms, but as an answer to some particular environmental conditions, amoeba produce chemical signals which cause the aggregative behavior of individual cells, and finally they form a motile multicellular organisms, which is called a slug. The slug migrate in search of the place for saurocarp formation. The transformation to saurocarp include differentiation to the stalk, which made of cells sacrificing themselves, and head, containing encysted amoeba, which finally disseminate in the environment to give rise to the new generation of independent unicellular amoeba. This cycle includes multicellular stages, cell-to-cell interactions, cell differentiation, and even formation of epithelia, very similar to those of multicellular animals. Probably it is the most complex example of aggregative multicellularity. In the conclusion, we need to emphasize that both clonal and aggregative multicellularity has evolved independently in multiple times in the course of the evolution in eukaryotes.